So I have the honor of introducing our speaker, Professor David DeMets. He is the former chair of the Department of Biostatistics and Medical Informatics at the University of Wisconsin at Madison, which he founded the department. But before that, he spent 10 years at the National Heart, Lung, and Blood Institute at the NIH in the United States, where he was chief of the Biostatistics branch. He joined the University of Wisconsin after that and founded the department, which he chaired. He is well known because he has co-authored 4 texts on fundamentals of clinical trials, on data monitoring committees, and the practical and statistical methods for clinical trials. He has served on numerous NIH and industry-sponsored data and safety monitoring committees in diverse disciplines. He's also been the president of the Society for Clinical Trials, elected fellow of the International Statistical Institute, the American Statistical Association, and a member of the U.S. National Academy of Medicine. He has served on review panels and committees for the Food and Drug Administration, the NIH, the CIHR, and the Institute of Medicine. He has broad research interests, and I had the opportunity to learn about them the last few days. But we all know him for the design, data monitoring, and analysis of clinical trials, especially large phase 3 randomized clinical trials. He's well known worldwide for developing the DeMets alpha spending function and sequential statistical methods for monitoring interim data for early evidence of intervention benefit or possible harm. I can't go on over the more than 100 very influential trials that he has had the opportunity to contribute to, either in the design, interim monitoring, or analysis of the trial final results. He is well known throughout, but a little less known fact is that despite serving on many of the data safety monitoring boards for PHRI, he has never been to Hamilton until today, or this visit. This is the first time he's coming to Hamilton. He always met at the Toronto airport for some of the DSMB meetings. We had a wonderful time the last 2 days as 2 Americans, and Salim talked about having the Pogue speakers be US people, but we had 2 Americans exploring how influential Hamilton was in repelling the invasion of the United States in 1812. So without further ado, I'll have David talk about data monitoring committees, past, present, and future.
Well, thank you for that introduction. It's a real honor and a pleasure to be here. When I got the call from Liana about possibility of coming, I know I had the opportunity to work with Janice Pogue for a few years, and clearly 2 things came to my mind when I thought about her. Well, first of all, she's a wonderful statistician, as you all know, and she's also a wonderful person, as you all know. But as I thought about her, 2 themes stuck out in my mind. One was the issue of statistics and research integrity, which I spoke about yesterday. And clearly, it was to me, it was overwhelming that I could have absolute confidence in Janice when she did something. And the second was monitoring committees, because that's where we worked together the most. But at any rate, I had great respect for her, and I understand the loss you felt when she was passed away. So anyway, I'm delighted to be here. I have to tell you, I'm on a dual mission today, during this evening. I want to share something about the impact of data monitoring committees, and I'm also on a recruitment mission, and you'll find out at the end why I say that. But at any rate, without going further, oops. So this issue of monitoring, it's got a long history, and maybe people have not taken the time to read this, but the Declaration of Helsinki in 1964 said, you can't just do a trial for the fun of it. You have to have a process in place that monitors the risks and assess the risk-benefit ratio. And when you have definitive proof about benefit or harm, you need to stop. It's not fair to just keep going because it's interesting. So for a very long time ago, we've had this mandate about clinical research. There have been many conferences and so forth, but probably the most influential for me was what we call the Greenberg Report. Bernie Greenberg was the chair of biostatistics at UNC and was asked to chair a committee that was commissioned by the National Heart, Lung, well, I guess it was called the National Heart Institute at the time. We now call it the National Heart, Lung, and Blood Institute. But there's a lot of clinical trials that were being thought about, considered, and they decided we needed to get a plan as to how we're going to do this. And so Bernie Greenberg and his committee were commissioned to write a report as to how that should go. It was shared in 1967. It was never published until 1988. When I joined the Heart Institute in 1983, you had to read the Greenberg report. That was your homework about how to proceed. So it's a very influential report. If you've not read it, you should. What did they recommend? Well, they recommend a lot of things, but the one thing that's important for this evening is you need to develop a mechanism to terminate a trial if the question's been answered, if it can't achieve its goals for whatever reason, something unusual turns up that has made it irrelevant, or the hypothesis that says no longer relevant, you've been scooped perhaps by somebody else. And they started out with a policy advisory board for the coronary drug project, and quickly over time they realized that was too big a group to deal with monitoring a trial. So they formed a subcommittee essentially, and that launched the history of data monitoring committees. So there's lots of reasons why you do this. It's ethical, as I just shared with you. It's scientific. You don't want to put a lot of money and energy into a trial that's not going anywhere or is not going to get an answer. And of course, there's economic reasons. You don't want to waste precious resources, including dollars. So it's quite a compelling reason to do this. So about, I don't know, maybe almost 20 years ago, a group was formed called the Clinical Trial Transformation Initiative. It was a joint effort between the FDA, Duke University, academics, institutions, and sponsors. And they would tackle certain problems that were bugging everybody. But among them, they said, what about this issue of data monitoring committees? So they formed a task force to look into this. And I was one of the founding members of City, by the way, but I was also involved with this particular project. And we realized that there's a lot of trials that claim to have a monitoring committee. Perhaps not necessary, but there they are. And so I wouldn't want to stake too much on these numbers, but roughly speaking, there's 120,000 trials in clinicaltrials.gov, and probably 30% of them claimed to have a monitoring committee. We said, where are all these people coming from? I've been around for 50 years in this business. And I don't think I know enough people to cover 30% of 120,000 trials. Well, it became very clear that we have a supply and demand problem. And the other thing we realized is there's no organized process by which to close that gap. Many of us get a little gray-haired, and that's just a long time, and there's not a process to replace this that's big enough and good enough to do the job. So they recommended that we have organize a training program of some kind. Didn't have the definite specifics, but in general. Well, the Society for Clinical Trials agreed to sort of help address this problem. And so with a zero budget, I'm used to that. We developed a training program that had three legs. One, we gathered some educational material. We knew that there'd been conferences that had been videoed. One at the FDA, one that we did with University of Minnesota at Wisconsin. There was a conference in Seattle. And so we retrieved those videos. They were, all day type workshops. So there was a lot of good material. People like Tom Fleming, Susan Ellenberg, Janet Wittes, and others contributed. So we retrieved those videos, and the Society for Clinical Trials agreed to put them up on their website. I'll mention how you get to them in a minute. We also said if we retrain people, we got to know where to find them. Because right now they use the word of mouth or the last committee they use, and so we don't make any progress in closing the gap. So we said we got to have a registry. If you can raise your hand, it's not a credentialing. We said no, we're not going to go down that path, but you can raise your hand and say I'm interested in a clinical trial. This is my experience, and please contact me if you think I can fit your needs. And then we said we need a mentoring program. You need to have a little experience at this, and there's been a lot of discussion about how to implement that mentoring program. But we have just actually published a paper with Frank not Harold, Rockhold, appealing to industry to buy into this program, this mentoring program. We'll see how it goes. So I'm not going to go into this in great detail because we're going to talk about other things a little bit. But the website is up and running. It's been running for a year now. And I think Hannah was on duty when we put this up. Thanks for the society for supporting this. But you can go onto this website and get probably a dozen or more hours of lecture on various relevant topics. And the registry is also up and running. I didn't check what the membership is, but we do need to populate this registry. It's a volunteer only, and you're up to yourself to keep it up to date. But anyway, we can come back to that. So that's the commercial, and I'll come back to why I put that commercial in later. So the first clinical trial. So I joined the Heart Institute in 1973 January, and the trial that was ongoing was the coronary drug project. It's where I learned most of what I know about clinical trials. I learned from reading about the coronary drug project. It's an amazing study. If you've not read it, I suggest you find the articles that talk about the coronary drug project. It was a trial studying lipid-lowering drugs. It had five drugs. Mortality was the outcome. It began in 1965, and it actually, the 5 arms, it terminated 3 of them early. 8,000 patients. It's quite large for its day, and still large today in some ways. But it stopped the high dose and low dose estrogen groups for increased cardiovascular events. It also stopped the dextrothyroxine arm for cardiovascular event risk. It continued the clofibrilate and the nicotinic acid arms to the end. But in that process, we learned, one, how to run multi-center trials a little bit. We also learned about designing them and monitoring them. So the authors or the founders of that trial really provide us a great roadmap about lots of issues about analysis and so forth of clinical trials. That after that trial got going and we had its own monitoring committee, that model sort of spread across the NIH, slower than you would like, maybe, but it eventually made its way across many of the institutes, and that became the standard of how we do things. And when Salim and I were at the institute together, that's kind of how things were done. So far, so good. But we also know, I always use 1990 as the benchmark, but about then, the NIH budget was pretty sad. There were lots of products out there to be studied. And so industry stepped forward, and the FDA also got involved to design trials with monitoring. The International Conference on Harmonization provides some guidelines. Interestingly enough, if you looked at the 1989 FDA guidelines about trials and monitoring, there's one paragraph. That's it. They to admit that monitoring exists, it goes on, but they provide no details, no guidelines, nothing. But the International Conference on Harmonization did provide some guidelines. Later, the FDA provided a specific guideline, and it was initial release in 2001. It was finalized in 2005. Interesting enough, there's only one situation where you're required, and that's when you have a waiver of informed consent, but we can talk about that. So this is the model that I live in terms of clinical trials. It's oversimplified, of course, but there's a regulatory agency that keeps an eye on things with a sponsor, which you need, a steering committee which produced the protocol, hopefully with some independence from the sponsor. But then there's 3 components, which and of course there's clinics and where the patients come from, but the data monitoring committee, independent monitoring committee, a statistical center that does the analysis for the monitoring, and a place where the data gets managed. I'll call it a coordinating center. Sometimes a CRO, sometimes a sponsor itself, sometimes an academic unit. So that's kind of the world that I live in. It's similar to what NIH does. In NIH trials, sometimes the statistical center and the monitoring committee or the coordinating center are merged in some way. But the reason this is such an interesting area is that we have responsibilities to several stakeholders. Patients, of course, your job is to protect the patients as best you can. Study investigators put their trust in this committee to keep an eye on the store so that things don't get off the tracks too far. Sponsors, of course, they're paying the bill. They have a lot of investment. You all know that you have to keep in touch with your local IRB. They want to know what happened, how often you met, and so what were your recommendations. And the regulatory agencies also have a responsibility. They pay less attention to the details up until recently. They've now suddenly gotten quite interested. But, you know, as I told them many times, if something goes wrong, they're going to get the blame. So they better pay attention. And they're just, but up till now, they haven't really understood too much of the details of how a monitoring committee operates, but we're getting better at that. Not every trial needs a monitoring committee. Salim and I have talked about that in other settings, but there's some places where you need to have a monitoring plan. That came out of another sort of problem that I won't go into in detail, but a trial that went off the tracks. The NIH policy has been, I think, that all trials have to have a monitoring plan, and most phase 3 trials are required to. I don't know if that's still the rule, but at least last time I was involved, it was. And now the FDA have those guidelines. So we have some guidance as to how we should set this thing up. Membership, who gets to do this very special job? Well, no one person, in my opinion or experience, could do this job by themselves. It's too much. So you need people with the clinical experience about the population that you are studying. You need some basic science. You understand how the mechanism of the drug or the device works. Clinical trial methodology, biostatistics, epidemiology, medical ethics, and so forth. And sometimes, not always, you have a person with some regulatory experience and or a position, but that's not regular. Experience is essential. One reason why I started paying attention to this gap problem, I was showing up at monitoring committees, and the chair would say, "I've never done this before. What do I do?" That gets your attention, and I really said, "Hold my hand. I'll help you." But that's not how you want to run the shop. So we have a problem of not having enough experience. Usually, monitoring committees have 3 to 7 members. You're getting bigger than that, you got a management problem. Less than that, you can't afford to have anybody mess. We try to get monitoring committees which have independence of the sponsor, the investigators. And there's all kinds of conflicts of interest. People think about money, right? That's constant. But there's if you're taking care of patients in the study, but you got a conflict. I mean, how can you take care of a patient when you're trying to figure out whether the drug works or not? Maybe there's a competing trial, and that sometimes happens because the pool of investigators is small. An intellectual investment. I've been involved in trials where people on the committee had spent much of their career developing this field or this device or this class of drugs. Well, they have a stake, and maybe doesn't make them ineligible, but at least you should know where they're coming from. And the other thing is it's not possible to avoid all conflicts. I mean, it's just you can't do it. If we have any expertise, we have a conflict. Maybe not a killer, but we have a conflict. We have to sort of divulge that, share it, and we do that. So what do? Well, we give a pretty good annual or semi-annual physical to these protocols. We look at data quality, timeliness. Are we recruiting? And usually recruitment is one of the first things that goes off the tracks. Can you get the patients you want in sufficient numbers? Check on eligibility and whether comparability, whether the randomization of the processes works. Of course, it's obvious you look at the primary and secondary outcomes, but you're also going to take a good hard look at the toxicity and adverse events, sometimes pre-specified subgroups. And for me, it's always important to look for internal and external consistency. I always say that good news is usually consistent and bad news is usually consistent. And if it's not consistent, be careful. You got to find out why that's the case. So monitoring committees can do a few things. They can recommend the protocol continue as planned. That's the usual case. They could say make some minor modifications to the protocol. Generally speaking, they don't like to make major. This is not their protocol. They're monitoring it on behalf of these stakeholders. But they may say a few things that need to be fixed up, or they may recommend terminating the trial. Why would you do that? Well, maybe you have serious toxicity. It's just not ethical to continue this trial in this population. Or maybe once in a while, you're lucky and you've established benefit. That doesn't happen often, but it does happen, and you got to be prepared for that. We talked earlier today about utility. Sometimes trials just aren't going anywhere. And that doesn't mean that you quit, but you better have a good discussion about why you want to continue this trial if it's not going to achieve the goals you set out. And sometimes it's just too messed up. You can't fix it. The wisest thing is to close shop and go home. So it's a very serious business, this process, and the decision process to come to a recommendation is complex. I just said you check all these items, and what you really want is everything lines up just perfectly. Then the decision is easier. But often the things don't line up exactly in the same direction. So now you have a very lengthy discussion. Something that has come up in some trials is, okay, you got the current patients in the trial, you got to protect them. But how about the patients that might be coming into the trial in the future? What for them? And how about the patients who aren't in the trial, but for whom this therapy might be applied? If you're not going to have an impact, you're not going to change practice, then why would you want to stop a trial? And you're not going to gain anything because no one's going to change their practice. So all these sort of non-statistical issues exist. There are statistical issues, and we're not going to go into that, even though that would be entertaining for me at least. We're not going to do that today. But there's lots of complicated issues, which is why you need a well-trained and experienced data monitoring committee to handle these things. It's not just looking at a 2x2 table and seeing what the p-value is. If you're going to be on this committee, you have to be willing and able to be confidential about the emerging results. That doesn't always, it happens to be the case. That's true most of the time. But every now and then you hear about a case where somebody inadvertently or perhaps inappropriately leaked interim results. Well, what happens? There used to be a saying in World War II that loose lips sink ships. Well, loose lips in trials can affect recruitment, compliance, outcome assessment, or even trial support. I have a case I could share with you. I won't have time. That the fact that there was some sense that the trial wasn't going well, the sponsor pulled the plug without actually seeing for sure what the results were. So you got to agree to that. That's absolutely essential. Only a brief comment about this issue of liability and contracts that we signed to do our monitoring committee work. We are liable, especially in this age that we live in, what we do, just as much as physicians are liable for their practice. And unfortunately, the contracts that I get to sign by serving on a monitoring committee have language that's totally inappropriate. The contracts are often off the shelf, have nothing to do with clinical trials. And if you don't pay attention, you might actually sign these contracts, but they will often have language that says, I, Dave DeMets, will indemnify company X. Well, so far, I don't think I can do that. Maybe there's still hope, but right now I can't do that. No, it's going to be the other way around. The contract needs to indemnify the DSMB. And sometimes the language says, oh, we'll indemnify you unless you are guilty of negligence. And I learned this from some lawyers who co-authored papers. Is that how your car insurance works? If you have an accident, you're negligent, I'll buy it. But don't you expect car insurance to pay for damages? Does your malpractice insurance work that way? So we have to have language that's appropriate. So there's papers written about that. And if you want to talk to me sometime offline, I'm happy to talk about that. But it's a serious issue. It doesn't happen often, but it has happened. The reason I know that is because I've been asked to be an expert witness. And I know a friend who spent hours defending himself in a case like that. Okay, interim analysis. Well, I said it's compelling that we do it. The Helsinki agreements require it, suggested, and so did the Greenberg report and many others. But here's the problem. This is the coronary drug project. One of the arms was clofibrate versus placebo. These are the mortality results over time in the final analysis. There's no difference in those two curves, right? So you wouldn't get too excited, but, oops, wrong direction. If you were to plot these results over time, you'll see something that we really need to pay attention to. This figure is laid out in the following way. On the x-axis is time in the trial, and on the y-axis is the standardized statistic. Take the difference divided by the standard error to get the standardized statistic. 2 or bigger gets people's attention. So I've drawn 2 parallel lines at +2 and -2. And you notice that this, as you track this study over time, on several occasions, it gets very close or slightly over 2. Yet we know at the end, there was no difference. So this is the dilemma that the statisticians have to pay attention to, and everybody, because you can easily get misled, especially early in the trial when the numbers are small. Small numbers, I call it the law of small numbers. They're not very trustworthy. and so you gotta pay attention to this. Well, this was provided by Paul Kenner, by the way, who writes a lot of the methodology papers out of the coronary drug project. Very, very smart guy. Well, I'm not again going into all the details of what we can do about this. I had a lot of fun developing methodology in this area. So there's more than one way to protect yourself from making a mistake. And so these are just some of the methodologies that are out there. You might recognize some of their names: Villalpito, Pocock, O'Brien, Fleming, Land, DeMets, and so forth. The literature is very rich about this. Now, when I started this in 73, the literature was very small, but we We spent a lot of time in the 70s figuring out what to do about this. I'm going to try to explain this slide simply. These are three possible boundaries that you might use to interpret these interim test statistics. So I don't know, does this thing work as a pointer? Maybe not. How do I can do this? I can use it. So okay. Yeah, well, I'll just use, oops, what happened? Yeah. So this boundary here is the boundary that Pogue published in 1977. This is the Hay-Biddle-Pito boundary, at least in its original form. And this curve boundary is the O'Brien-Fleming boundary. Now, you have to pick a boundary that suits your needs. There's no right one. I know there's a preferred one here, and we've talked about that earlier today. But there's more than one way to protect you from making a mistake of a false claim, false positive claim, of benefit or harm for that matter. So I'm not going to go as to which ones you should use. That would be another whole discussion. But I am going to refer to this kind of picture later in my talk. So you need to specify what kind of monitoring guidelines you're going to use in your trial. You can't look at the data and then draw the line, right? You got to draw the line and see whether you cross it or not. So this is, I said, a whole topic for a full hour lecture at least. There's also another technique which we call conditional power. And then in any interim point in the trial, you say, what's the probability, given the results I have so far, will I have a successful positive study or perhaps negative harmful study? For those of you who are not statistically inclined, you can think of this as a golf tournament. You played the first two rounds, and now the question is, how many strokes are you behind? If you're 20 strokes behind, maybe your chances of winning this tournament not so good. If you're 3-4 strokes behind, maybe you have a good. So you can calculate the probability of winning given where you are, because you got some part of your trial left, half of it, let's say. So it's a very handy technique. It's not that hard to compute these things, and we do that all the time. Oops, I keep going in the wrong direction. Now where am I? There we go. So I plotted the same kind of picture, and what these green lines are is if you cross using plotting those statistics, cross any one of these boundaries, you are having a small chance of winning, being positive in this trial. You can pick different probabilities. If you want to be very conservative and say the conditional power is less than 10%, okay, that's pretty conservative. Or maybe you don't want to be that conservative, you can pick 20% or 30%. That's arbitrary. I can't tell you what you should do. It's a question of how much of a gambler are you? But these techniques, I can tell you, both these or variations of them are very common in assessing interim results. So I want to spend the rest of my time sharing with you some scenarios that I think each one of them challenge the monitoring committee as to what they should do. What's the right thing to do? Who knows what the right thing to do is? You got to do something. So I want to share with you a scenario where the benefits are slowly emerging or rapidly emerging, where negative results are slowly coming out or rapidly coming out, or a trial for which there's no benefit likely. So we're going to show each of those very simply because of time. So the first trial that I actually implemented a sequential boundary of the kind on that one graph was the beta blocker heart attack trial. It was a classic post-MI study, randomized, placebo-controlled, 30 to 100 patients in those days. That was big. And it was post-MI patients who were 5 to 21 days out from their MI. So we were going to test a drug, this beta blocker called propranolol. The results, they came in over time. We started in May of 1979. And over the next couple of years, we got these results. You can see the number of deaths in the propranolol group and the placebo group, and on the right-hand side is the standardized statistic generated by the log rank test. So as I said, 2 or bigger would be of interest to people. And you notice that this trial continued on a lot longer than after we crossed a rank statistic of 2 or bigger. Well, how in the world did we justify that? Well, early in the trial, I proposed that we use the O'Brien-Fleming boundary. I believe it was the first use of the O'Brien-Fleming boundary, at least in a major study. And so what you see here is the results plotted over time. And you notice that in October of 81, it crossed the Bryan-Fleming boundary by a substantial amount. And so we said, okay, this is good enough. We have a year about to go. And so the committee recommended to the Art Institute that they stop this trial early. Stopping trials early in those days was really kind of new. The FDA came down with great interest and great worry about why did we do this? Because they knew they were going to have to review this study shortly. But at any rate, this O'Brien-Fleming boundary said this is not likely by chance alone. This is probably a real effect. The time to event curves were quite separated. And I'm not going to go through the whole thing. I mean, we got a report that's half inch or more thick to go through. But I want excuse me, I'll share with you just a couple tables. Consistency, well, it didn't matter age, sex, or race. The results were still in the same direction. That's good. I keep messing up here. I guess that's all I showed you. Anyway, there's a whole series of slides that show the side effects were in the right direction. The subgroups were in the same direction. The other secondary outcomes in the same direction. So everything was lining up. We were all happy. We recommended it, and it got approved. So it took a while, as I showed you, for that result to emerge. It didn't happen, in the first 6 months. But a different scenario. So heart failure was and is still one of the major problems in cardiac disease. And when this trial was designed in the mid-1990s, it was common wisdom that you did not give a beta blocker to a heart failure patient. It seemed like it was doing things that you didn't want it to do. It slowed the heart rate, lowered the blood pressure, all those things. But thanks to a couple of Swedish investigators and perhaps others who persisted in testing beta blockers in their heart failure patients, enough evidence came out that this was a worthwhile study to do. But nevertheless, this was a considered kind of a risky deal. And so the FDA, by the way, was so nervous, they made the monitoring committee meet every month because they thought for sure there was going to be trouble. And so we met. You talk about the workload of PHRI. Well, we had to put out a full report. Now, and we had also, to get into the study, you had to have a 2-week run-in period. We randomized almost 4,000 patients. Big study for that day. And it was the classic heart failure patients of class 2 or 3 heart failure, mostly men, and at least in this study, with low ejection fraction. So a good population. Well, contrary to expectation, we got a result that I would say shocked some parts of the cardiology world, a very highly significant benefit, despite all the worries, And so it was the first of a few trials that tested beta blockers. If you were to have followed the trial's progress as I set up, you'll find that this study, oops, maybe I'll get this right. You start out at 0, you have no data, and you have no results, so you don't have any statistical evidence. But the first serious analysis we get, the value is a z-value of 2.55. Now that's getting to the point where you start paying attention. We had agreed in the charter that we would not stop this trial because of all the worries until we got at least half the patients, half the follow-up. So there was enough long-term data that you could see what happened. So we went on for another round. And in the next round, we had this z-value of 3.8. And that's pretty substantial. These monitoring boundaries, this is a variation of a Haber-Lobito. This is a variation of a Pogue. They don't have to be symmetric, by the way. That's another whole discussion. But we had met our criteria. We had enough data that we understood the short-term effects and longer-term effects. And we had probably the second most largest z-value that I've had in my career. So we recommended that this trial be terminated early. For all the different outcomes, these are the so-called forest plots. We plot the hazard ratio and its confidence interval. One is so there's no difference. So it's all to the left of that line, which says beta blockers are good. So pretty consistent results for different cardiovascular events. If you looked at subgroups, classical subgroups that any cardiologist would be interested in, again, all to the left. Again, consistency. And so we felt that it was a very good recommendation to stop early. Now, I don't have a slide for this, but I have another slide that looks like this by geographic region. I guess I should have included it. But it turns out everything was great except for one place, the United States. Mortality was flat on, hazard ratio of 1.0 something for mortality. For cardiovascular mortality and hospitalization, it was in the positive direction. Well, this caused a huge problem for the investigators and the sponsor. And I even wrote a paper with my good friend Hans Weil about this. But at any rate, it was consistent everywhere but one place that was the politically critical, that is the US. And so the FDA never did approve it in the United States for mortality. They wiggled out by approving it for cardiovascular death and hospitalization for heart failure. So they approved it, but not for the indication that everybody else got. I think it was an erroneous investigation, or at least an issue that was raised. And so as we wrote in our paper. And then it turned out other related beta blocker trials came along and said, no, that was just by chance. But nevertheless, the fact is that, you're going to find something by chance alone. So the second comment I'd make is how do you share these kind of results with an investigator team who already is a bit gun shy because it's a beta blocker in heart failure patients? Well, so we met in Paris, good place to meet. And we met in a room and we said, we're only at monitoring, we said, Desmond Julian, by the way, somebody was the chair. We said, we're only going to tell you one thing. Overall, you have a mortality benefit. Do you want to know more or not? And we're going to leave and you can talk about this. So we came back in, I don't know, 20 minutes. We said, well, what do you think? You want to know more? I said, yeah, we want to know more. So we gave them more. And then they got some of the results we just saw. And finally we said, do you want to know everything? I said, yeah. So we gave them the monitoring report. It was a way, why did we do that? Well, if they disagreed because of the context that we should stop early for a mortality result of this kind, then at least we hadn't, it was only the senior leadership of the trial, you protected the integrity of the trial as best you can in that situation. So how you reveal results to investigators is something you got to think through very carefully. Anyway, so those are an example of a slowly approaching, evolving benefit and a very fast approaching benefit. Now I want to flip around and look at the other direction. This trial, the cardiac release suppression trial, was one of those landmark trials that you never forget. The final results were published in 1991. Cardiac arrhythmias are associated with sudden death. We have drugs that will suppress arrhythmias. We got them, approved them, and began to use them. But CAS was designed to ask the question, do these anti-arrhythmia drugs actually reduce sudden death? And so we had alpha spending functions, and I didn't get into that, but it was designed basically with a one-sided 0.025 criteria. So we meet in September of '88, and these are the results that we were shown. Monitoring doesn't know what A and B are. We don't know what A and B are. But it was expected that this would be a positive result, but we were blinded. And so we said, well, it doesn't meet the 3.18 minus and 3.03 are the boundaries we have to exceed, and we don't exceed it, even though it's an interesting direction. And we're very early in the trial, only 13% of the information. So it's pretty early in the trial. You might not want to buy it, but something interesting. And if you plot the results on the boundaries that we had used for this trial, you notice we're close, but not quite. So a couple of months go by, and we get this alert from the statistical center saying, we need to share with you that this is in the wrong direction, contrary to popular expectation. So the first reaction, well, somebody made a mistake. This is not the way it's supposed to happen. So we spent about 2 months going through this fire drill. Was there error in the packaging? Was there error in the distribution? Was there an error in the programming? A whole long list. It took us probably 6 weeks to go through. First of all, I had to get the data in-house, clean out the pipeline. but everything was consistent, and it was consistent in the wrong direction. And so we had an emergency meeting in March of 1989 in Seattle. I called the Sleepless in Seattle meeting. And so by the time we got together, a couple months down the road, the results had cleared the boundary by a substantial amount. And we made a recommendation that this trial needed to stop for harm. And so there's the plot. So this happened very fast, contrary to expectation. And so it was on a Sunday afternoon, we made this recommendation. We called Bob Temple at the FDA and said, we have a problem. Houston, we have a problem. And so they set out a physician alert, and you guys know more about that than I would, but it set off, ripples throughout the cardiology community that this therapy, which was supposed to be very effective, in fact, was causing harm. So things that happen fast. What are the lessons I learned? There's the mortality curves, or time to event curves, survival curves, put it that way. So what lessons we learned? Well, first of all, lesson number one is I will never, ever agree to a trial in which the monitoring is blinded. I mean, this was a disaster in some ways. Second of all, you got to be prepared right away. You can't wait till you get around to it. Your operation has to be ready to go on day one, essentially, because this happened like within the first 10, 15% of the trial. So you can't wait around. You want to minimize the pipeline because the monitoring is going to worry, and we did. What is it we don't know? It's already happened. And if the data are 6 months old, that's not helpful. 1 month old, yeah, maybe we can live with that. So you have to have that pipeline flushed. Double check randomization. I shared it at lunch today. I've had 2 examples where the randomization code that was sent to us to prepare the report was wrong. It was random. If you generate random numbers, the computer algorithm has a seed. It's built usually into the software. You run it once, you use one seed. You run it again, you get a different seed. You get a different randomization sequence. Well, what happened, we learned later, is that the statistician who ran the randomization code and gave it to the pharmacy that made the packages and sent them out had one list. When they sent it to us, he reran it again. He didn't send us a copy. He reran the program, so we had a new randomization scheme. So now we have a randomization scheme which is totally bogus. So double check randomization. Never design a one-sided hypothesis. This was supposed to work. And so the story, if you read the paper, it's a little schizophrenic. They talk about the one-sided hypothesis of reducing sudden death. And initially it was at a one-sided 0.05, and I said, no. That's not any good. We can't have that. That's too weak evidence. They said, well, how would you accept one-sided 0.025? I said, sure. Just give me the other 0.025. And I want to use it for the wrong direction. I did that not knowing anything about the results. I just said, good old DeMets stubbornness. So we put it in, it says it's an advisory boundary. Well, sure enough, we crossed the advisory boundary for harm. Then there was a big debate in the paper. Can we claim harm or just say it didn't work? I said, if you say that, you make statistics look stupid. This is clearly the wrong direction, and I would say it's significantly in the wrong direction. So another trial which went in the wrong direction was one of the first trials I did with industry called Promise. Turned out it didn't show any promise, but milrinone was approved for the IV use of high-risk heart failure patients to improve cardiac function. We randomized 1,000+ patients, and it was stopped early for more deaths on milrinone than on placebo. And if you look at the plots, and by the way, that result at the end was consistent across ejection fraction categories and New York Heart Class categories. So it was pretty consistent, which helped us understand what was going on. But if you were to plot those results over time, you can see that the first meeting, the z-value is in the wrong direction. Meeting 2 is not quite in the wrong direction, then it comes back again. And finally, after meeting 4, we're down here where we cross these boundaries. You say, well, why in the world? And and I didn't plot this, I should have included that slide, but you cross the futility boundaries somewhere about in here. You're not going to recover a positive result. Well, why hang on so long? One can debate that, but one discussion point was this drug has some benefit, maybe not great benefit, but it does improve cardiac function. So we felt we had to rule out is it just not effective for mortality, or is it in fact harmful? If it's neutral, maybe the drug has a future. So this debate went on for a while, as you can tell. And so that's another thing you have to figure out is, if I stop it now, what are the consequences? What's the impact of stopping it? So if we stopped it early and say, well, it just didn't work, people say, yeah, but I like it. It makes my patients feel better, makes them function better. So you got to go through that discussion. The lessons, yeah, I need to root out neutrality, harm from mortality. So this was such an agonizing experience that Desmond Julian, Stuart Pogue, and I wrote a paper called The Agonizing Negative Trend. And the idea of this is that if you're testing a new drug out of the box, you might bail out a lot earlier if it doesn't look so hot. But if it's a drug that's in use, that's popular, like hormone replacement therapy for women, it's already popular. You got to have strong evidence to turn that ship around. So these are non-statistical factors that you have to consider, and which is why I said you need a well-trained, experienced monitoring committee. Physicians' health study is my next to the last example. This is an older study testing the use of aspirin to reduce cardiovascular mortality and fatal, non-fatal MI. And it was a factorial study, beta carotene and alpha tocopherol. That was the hypothesis. It was double blind, group sequential, and all the, I would say, the right pieces. But it went on and went on and went on. And so after a certain period of time, we debated what to do after a 15-month debate, you might say. First of all, the mortality result is probably the most underpowered trial I've ever run into. We were expecting 800 deaths in that amount of time, and we were getting a little over 80, 1/10 of what we were expecting. The mortality question was so-called dead on arrival. But we were getting this interesting fatal and non-fatal MI result, a relative risk of 0.53, highly significant across boundaries. But to counterweight that, we had a stroke problem. 10 hemorrhagic strokes was 10 versus 2. And on top of that, physicians, knowledgeable physicians, if they had an MI, they were switching over to aspirin. We had a huge drop-in problem. So you got all these factors stirring in the pot. And we debated for 15 months, and finally said, this non-fatal MI result is something we need to share. And we're probably not going to ever have enough data in stroke to settle it convincingly. So we recommended termination, and the PI, I would say somewhat reluctantly, accepted it. So I've been criticized for this. I chaired the monitoring committee eventually, criticized for that, but I have to tell you, I have not lost any sleep over this decision, because I did an informal survey. Anybody I knew, maybe you can ask Herschel, if you were in this study, did you take aspirin? I don't know what you said, but many of the physicians I asked said, yeah, we should have stopped because that's an important result. So I said, I'll go to bed and sleep well. But it is a debatable issue, which deserves debate. So when you looked at all the other results, they were very consistent. The MI results showed a benefit. And the stroke results, as I said, the total strokes are in the wrong direction, especially hemorrhagic stroke. So the issues that we had to fuss with were low mortality, a very positive fatal, non-fatal MI, a stroke concern, and crossovers. So there you go. Get your committee together and let's have a discussion. Now I want to end with this one because coming here, I have to talk about SOLVE. I was not part of SOLVE, so I'm hoping to get some feedback from the locals here about this study. But what is SOLVE if you don't know? Well, it was a trial of enalapril with patients who had low ejection fraction. There was 2 trials under one umbrella, a treatment trial for patients treated for heart failure with a low ejection fraction. And there's a prevention trial, patients who had low ejection fractions, but weren't yet, as I understand it, being treated for heart failure. So you got 2 trials running in parallel. The primary outcome was mortality. And then there's these other secondary outcomes for prevention. So as far as I could tell in reading it, it's pretty well designed, pretty well thought out. But yeah, it was designed well. Of course it was designed well. Salim was involved. And so there you go. They're looking for about a 25% reduction, 5 years of follow-up, 3 years of recruitment. The sample sizes were 2,500 for treatment because the event rate's higher. You can get fewer patients. Prevention trial was 4,600. Now there was some adjustments along the way. We won't get into all that discussion because it's complicated. But what I want to show you is a lesson that I learned from reading about Solved. And I think it was Janice who actually gave me these results. I suspect, I don't know who else would have given them to me. But anyway, at 52 months of follow-up in the treatment trial, those are the time to the survival curves. And here is, if you plot the data over time, and I show you the final results, it took off, backed off, took off, tiptoed across the boundary, and tiptoed back under it again. I would love to have been a discussion, a fly on the wall, listening to this discussion as to what they actually said and thought, because it had to be very interesting. And maybe, Salim, you can fill in the gaps, I don't know. But the reason I share this is because it's a good lesson about how variable interim results are. Early on, you just can't trust them. It's small numbers, and they can change very quickly. It doesn't take many events to change it. And here it eventually settles down, sort of in the right neighborhood. This is the prevention trial. Again, the longer the trial goes, the more interesting the results become. But if you were to plot those results over time, you can see that it zigs and zags all over the place. Early on, it looks like you're going to have a winner very quickly. That might not have been believable, in my opinion, for a prevention trial. But there it is. So it didn't cross. And then it took a dive in the other direction. Eventually, it was setting at 52 months, it was setting slightly in the right direction, but not good enough. Well, fortunately for the investigators, and when you got, so the trial continued on, I guess it terminated at 55 months for the treatment trial. And you cleaned up the pipeline, got all the data in, everything was good. The mortality was good, death and hospitalization was good. So that was, I assume, a pretty solid positive result that you got. The prevention trial never got a mortality result that was good enough, but it had a really strong death hospitalization, heart failure hospitalization result. So in the end, everything came out relatively nice. But the issue of the wandering of these results over time is the most irregular that I've seen. So Solved taught me in reading about it that interim data can vary widely. Early trends can disappear quickly. And if you cross a boundary, and this is a discussion which I saw, it crossed the boundary by a little bit, and it came back. What was the discussion? My view, and I think Salim from this morning's discussion, we agree, you want to cross not by just a little bit, you want to cross by a lot. Why? Well, there's the pipeline effect, and you want to make sure that you don't fall back, because that's hard to explain. You stop early and the results aren't significant. How do you explain that one? So you want to cross the boundary by a substantial margin. So the reason I bring this up, in part, I said to honor Janice Pogue, who I worked with on several trials, but also to make an appeal to you guys. We need your help desperately in helping to close the gap. You guys are sitting on a wealth of trials. You have a wealth of expertise. You're perfectly situated to help train the next generation of data monitoring committee members, both clinicians and statistical. So my appeal to you is please step in. And I know you're doing some things already. Do as much as you can, because we really want to have, the window is closing for which we can get this training done. So we got a lot of material out there for you to use now. You can generate your own, you're in a position to do that. But your experience and being able to share the operations of a data monitoring committee with young people experience is always important. So you're perfectly situated, in my opinion, to do something really powerful. Anyway, that's my commercial, and I'm going to stop.